Feeling good, wanna feel great. Longevity starts, don't hesitate. Too much noise, what do you choose? Trendy hacks, conflicting news. Skip the myths, come hear what's true. From world-class experts straight to you. First and third Thursdays, 7 p.m. Science, health, and our team. Brain to heart, from food to rest. Live the talk on living your best. Thursdays at 7, we'll see you there.
How much does AI really accelerate drug discovery right now? Is it an order of magnitude? Is it twofold? Is it a myth? Where are we?
Yeah, so I'd like to frame it in terms of two areas where AI either is useful or could be useful. So one where I think we're already seeing the use case is AI as a tool for automation, for discovery, for improving the efficiency of research. The second half of this, and I think this is where people really think about AI in drug discovery, is AI predicting new drugs and predicting, what are the things we should be developing.
Welcome to the National University of Singapore Yongle Lin School of Medicine Healthy Longevity webinar. Thank you for joining us. In our last episode, you heard Dr. Ben Blue talking about the use of AI for drug discovery. You can watch the full episode on our YouTube channel. Today, Associate Professor Evandro Fang-Stavem is with us tonight. Mitochondria and dementia is really his research topic, and he will also talk about NAD precursors. Please use the Q&A function to send your questions. Tonight, we have Sharanya Yajaprakash with us, a final year PhD student, and she will moderate your questions. So she will introduce herself first.
Hello, my name is Sharanya Yajaprakash. I am doing my final year of PhD under Associate Professor Sridharan, who is the Research Director of the Healthy Longevity Translational Research Program. And my research involves identifying how learning and memory processes are affected in Alzheimer's disease as viewed through the lens of neuroinflammation. My background is in clinical medicine, so I was working as emergency medicine physician, and so a lot of the bedside techniques were new to me when I first started in research. But what drew me to medicine in the first place was physiology and how it alters and almost distorts into pathological conditions, and the molecular mechanisms involved in that process, and how we can manipulate those very processes to bring it back to its homeostatic state. So my perspective on how I view research has been shaped by my clinical background. So it's rooted in the problems we face in the clinical setting and how research can help develop answers to counter that. The story of Alzheimer's disease is a very old one, more than a century now, in fact. When Dr. Alois Alzheimer first met Mrs. August at the hospital he was working in at that time, where she was admitted because of her deteriorating mental condition, little would he have realized what that encounter would signify to medical science in the future. Because fast forward to a century later, and Alzheimer's disease is now the largest cause of dementia-related disorders in the world. And why is it that a disease that has been around for more than a century is still so critical? One reason is many countries in the world are facing a rapidly aging population. Case in point, Singapore. And aging is a predisposing factor for developing Alzheimer's disease. The second could be that even after a century of research, we still do not have a viable therapeutic solution to completely manage the condition. So it is imperative that we are able to develop a strategy to fully counter it, because as it stands now, once you develop Alzheimer's disease, then there is no way back. So Why is it that we still have not been able to develop a solution? It could be because of the complex pathophysiological mechanism of the disease that we still have not understood fully yet. But through the research over the years, one factor have emerged clear, and that is the prevalence of neuroinflammation. So if we can understand how neuroinflammatory mechanisms work in Alzheimer's, that may provide an answer. And for that, we need to understand the agents and elements guiding that process, and that will be cytokines, chemokines, and their receptors. So there is one chemokine receptor that have gained attention during the recent years because of its effect in learning and memory in other neuropathological states. But its effect in Alzheimer's is still quite obscure, and that is CCR5. So if we can understand how CCR5 affects learning and memory processes in Alzheimer's, that may help us bring us closer to a solution. Now, to understand learning and memory processes, what do we do? And for that, we need to understand the concept of synaptic and associative plasticity. Now, simply put, synaptic plasticity is the ability of neurons to change, to alter, depending upon their activity levels. They are dynamic structures. And associative plasticity is the ability we have to link weak and strong memory together. So using a combination of field electrophysiology, molecular techniques, and behavioral studies, in my research project, I aim to understand how synaptic and associative plasticity is affected in Alzheimer's disease, and whether CCR5 have any role in modulating these processes. This would help us develop a therapeutic solution via using CCR5 as a mediator. One of the most rewarding moments of research is when you hit upon that elusive finding or results that will help explain your research story. And the second most fulfilling moment is when you get an opportunity to present your research to your peers. So the feedback and comments you get can really shape how your story turns out. For me, I got such an opportunity at the recent Healthy Longevity and Medicine Singapore, Japan conference, where I was extremely fortunate to have been able to receive the Best Oral Presenter Award. This wouldn't have been possible without the tremendous support that I've received from my supervisors, Prof Sajid Kumar and Dr. Likwe, my wonderful lab colleagues, and my beautiful family who have been there through thick and thin for me. So I'm grateful for all of them for my research story and where I am now. Please feel free to connect with me via Outlook. I will be moderating the Q&A session later on, so please add in your questions and comments in the chat. Thank you all for listening, and see you guys later.
Thank you so much, Sharanya. Our guest today is Associate Professor Evandro Fang-Stavem, and we will stay in the topic of dementia. He is a molecular gerontologist at the University of Oslo, where he studies the molecular mechanisms of aging and neurodegeneration. His work focuses especially on how defective mitophagy, so in mitochondria, contributes to dementia. He is highly cited, and he publishes enormous amount of papers in leading journals and received national recognition in Norway for his contributions to dementia research. Evandro, you're not only a great researcher, but I will call you also a very, very good friend. Thank you so much joining the webinar today, and we are looking forward to hear everything what you do in your lab to really maybe even cure dementia and Alzheimer's disease, or even prevent it from occurring. Thank you.
Thank you so much, Andrea, for this wonderful opportunity. I'm very grateful to be here with all my colleagues. And I was very happy that Sharanya also works on Alzheimer's disease. Hopefully, our joint venture will help to tackle this disease very soon. And this is my conflict of interest. And first, I want to share with you a video.
She's looking at you.
I know. I can't remember her name. What's her name?
It's Betty in front of you.
Who are you talking to? This lady here.
Betty.
I'm Betty.
Well, she's Betty too.
Oh, are you Betty too? Okay. All right. We got that settled. Wait, now I'm gonna get a good night's sleep.
There you go.
Thank you.
And it is obvious that Betty has her memory loss and with a diagnosis of Alzheimer's disease. And as Shransha mentions, Alzheimer's disease is one of the most common dementia affecting millions of people around the world. And she also mentions aging is a primary driver of Alzheimer's disease. To enable us to understand the causes of Alzheimer's disease and to develop methods, we need to understand the mechanism of aging. And I got my training in Baltimore. I want to share with you this video, these slides with Niels Bohr and Åge Bohr, two Nobel Prize winners from Denmark. And their grandson, Wilhelm Bohr, was my postdoc mentor. And I worked with him for 6 years. And in his 70-year birthday, we proposed additional hallmarks of aging, in addition to the left 9 hallmarks of aging, such as genomic instability, telomere attrition. And for the new hallmarks of aging, we propose that compromised autophagy, among others, are new hallmarks, and which were also included in the 2023 hallmarks of aging. A very interesting thing is so our group majorly focusing on mitochondrial dysfunction and compromised autophagy, their contribution in the aging process and Alzheimer's disease. While many molecules linked to those hallmarks of aging, one named nicotinamide adenine dinucleotide oxidized form, it is reduced during aging and can contribute to many of those hallmarks. And this is a topic of my group. So this is a logo of our group. We're interested in the mechanism of aging, especially brain aging. And we argue when we're getting older, reduced garbage clearance contribute to different neurodegenerative disorders. And the region in red here is entorhinal cortex as well as hippocampus, regions important for memory. And with this in mind, we have 4 topics in our group. The first one is why NAD is reduced during aging, and how can we safely and pharmacologically reinstall tissue NAD level. The second topic is age-predisposed Alzheimer's disease, and we propose that reduced garbage recycling is a cause of the disease. We are also interested in using artificial intelligence to prepare our drug development. And finally, we are also working on clinical trials, including leading a phase 2 clinical trial. If we focus on the process autophagy, the garbage recycling process, it contains macroautophagy, microautophagy, chaperone-mediated autophagy. For macroautophagy, which is a normal one we hear in our most common research papers, and it is engulfed by this double membrane effect pore fused with acidic lysosome to degrade any subcellular components. If the component is a damaged mitochondrion, we name it mitophagy, and it is a key topic in our group. There's a protein called ULK1, which is important for the autophagy machinery, but also essential for the mitophagy pathway. If we go back to Betty again, when she got married at her 20s, she has had very good memory. She only started to complain of memory loss at her 60s and reduced her memory at her 80s. And the brain, if we look at the schematic model, when at her 20s, she had a very clear brain with healthy neurons important for memory storage. However, aging drives accumulation of the garbage, which can reduce the function of those neurons. And it's like the city, we needed to have a garbage recycling truck to work. Once it's used for a long time, then the garbage recycling engine worn out and does not work anymore. And our group aims to focusing on repair of the engine as a way to increase the garbage recycling capacity. There are many approaches, and we focus on including both non-specific like exercise, fasting, healthy diet, taking care of mental health, and most specifically, the development of specific mitophagy inducers. So there are 4 questions we would like to ask. The first one is why mitophagy is reduced in Alzheimer's disease. Second, if mitophagy is reduced, whether mitophagy restoration inhibits AD. And third question is how to safely induce mitophagy. This last one, any applications of those mitophagy proteins in clinical evidence. The first one is why mitophagy is compromised in AD. As I mentioned, we are interested in many autophagy proteins with one named ULK1 based on our proteomic and Western blotting results showing ULK1 is reduced. So how we quantify the process of mitophagy? Colleagues in the field use a mitochema reporter. This reporter has a green signal if the mitochondria are healthy and not degraded by mitophagy. Once the mitochondria are damaged by FCCP and some molecules, and you can see dramatic increase of red dots indicating high mitophagy levels. And we argue reduced mitophagy is a phenomenon of aging and a risk for many disorders, including Alzheimer's disease. But whether mitophagy is really reduced during aging, especially in live animals, the data are not clear. And with our colleagues, we used a 2-photon microscopy and worked with live mice. We indeed saw age-dependent reduction of neuronal mitophagy, as well as reduced mitophagy in astrocytes, which is another brain-supporting cell. So as I mentioned, our paper 5 years ago showing ULK1 was reduced was based on 8 patient samples. But whether this is really the case or it's just we are so lucky. To validate the changes of ULK1, we used the large human samples we got from King's College London, and we show a tau stage dependent reduction of ULK1 in the neurons of post-mortem brain tissues from AD patients. And ULK1 is also reduced in both serum and CSF samples from AD patients compared to control groups. And this protein is also reduced in healthy cohort without neuronal damage, even after a 4-year follow-up. So make a long story in short, we propose reduced ULK1 causes reduced mitochondrial homeostasis and increased A beta and tau pathology. To validate ULK1 reduction is a driver rather than a bystander, we generated a ULK1 overexpressed mouse model, and we saw reduced tau, reduced A beta, and increased brain memory resilience. Now we go to the second question, whether mitophagy reduction restoration inhibits AD. And just now, I already used the genetic approach to show you ULK1 induction can inhibit AD pathology. But how about small compounds? And for example, in our group, we used NAD plus precursors such as nicotinamide riboside, nicotinamide mononucleotide, a molecule with its precursor available in pomegranate, like uranexin A, to induce mitophagy. We saw increased mitochondrial homeostasis and increased neuronal survival, increased phagocytotic capacity to reduce extracellular A beta plaques, reduced inflammation, and finally increased memory in preclinical animal models. And our colleague in Norway is using NAD precursor to treat Alzheimer's disease, in addition to Parkinson's disease, among others. So then the third question is how to safely induce autophagy. I mentioned we use NAD plus precursors. We have been using AI as approach and identified compounds like K and R. We also use the uranexin A, a molecule can induce mitophagy, as well as a molecule available in passion fruit called alpha amyrin. And I want to go a little bit more details on NAD and alpha amyrin in today's lecture. So how NAD can induce autophagy? due to the time limit, I want to give you a summary. So we showed that NADs used by the NAD-dependent sirtuins to increase the deacylation of key transcriptional factor like FOXO3 to transcriptionally upregulate genes encoding proteins involved in autophagy and mitophagy, such as BNIP3, NIX, LC3, ATG12, among other molecular pathways. A few weeks ago, we have identified a new molecular mechanism on how NAD increase brain resilience using a cross-species approach. And we showed that a protein called REST, which is important for brain resilience, but it is reduced in Alzheimer's disease. With NAD plus precursor treatment, we're able to see increased RNA splicing of REST, enabling neurons to express more brain protective REST isoforms, such as including XO1 and XO3, and finally to increase the expression of neuronal protective REST isoforms. I don't know how many of you have watched the Winter Olympics. Norway, a small country of 5 million people, again as #1 for gold medal, and especially this Johannes guy who got 6 gold medals and has been a hero in the country. And for us to celebrate this, we just went to the snow and to enjoy this. And because in Norway, doing outdoor sports is a routine, so we wondered that to treat Alzheimer's disease, in addition to feed the mice with NAD precursor, if we also force the animals to go treadmill running 30 minutes per day, whether we can see any additive or synergistic effects. And the answer is yes, with a paper in preparation. And the last question is whether those mitophagy autophagy proteins can be detectable in CSF and blood and can be used as biomarkers. With our colleagues in Czech Republic, we collect cognitive normal control, MCI, and AD patient samples, and we show the mitophagy protein PINK1 was reduced from control to AD, while another mitophagy protein NIX was also increased from control to AD. And a protein TFAP, which is important for lysosome function, which was reduced from control to AD. And our study was selected as one of top 11 scientific discoveries in all the fields of 2024 in Czech Republic. I want to use a few more minutes to share with you regarding the benefit of NAD precursor to treat premature aging disorders. With my colleagues Morten Skybye, Knudsen, under the mentorship of Wilhelm Bohr, we work on a group of disorders called accelerated aging diseases, as exemplified by Werner syndrome. And you can see this lady was only 48 years old, but looks like she was at her 70s. Another disease is ataxia telangiectasia, due to mutation of the ATM gene, causing damage of Purkinje cells and shrinkage of cerebellum. So the kids are unable to work very stably. So we, based on over like 10 years of work, we propose that DNA damage or reduced DNA repair can cause a depletion of NAD+, and which can contribute to the development of those premature aging disorders, including Werner syndrome and ataxia telangiectasia. To tap, because all those studies are from preclinical, we wondered whether our preclinical studies can be translated to clinical benefit. Working with my former head of department, Hilde Nielsen, we treated 10, in Norway we have 28 kids with the AT registered, and we were able to recruit 10 kids. And we treated them with NAD precursor NR for 2 years. And we saw dramatic improvements in 9 of the 10 participants. And the important thing was our data were also repeated by the Netherlands as well as Austria. I want to share with you the result of one kit published in Austria. This was a 3.5-year-old boy. The doctor asked the boy to draw the line between the hand and the balloon, and you can see how worse performance was. And after 9 months, and this boy was dramatically able to improve his performance after treated with the NR. So we're very pleased on this result. Another disease is Werner syndrome. Wilhelm Bohr had worked with Professor Kotaro Kote in Chiba University in Japan. With 1 gram of NR treatment, they also saw dramatic improvements in some, although not all of the endpoints they were looking for, including increased repair of the skin ulcer area. I think in University of Singapore, there are also many colleagues who are distinguished researchers in the NAD field. And NAD has also been commonly used as a diet supplement. So there are many questions regarding how safe it is. If I have cancer, whether I can have the NAD precursor or not. Which NAD precursor is the best? To address those questions with our colleagues, including David Sinclair, among others, we wrote this expert opinion to give guidance to the field. Another molecule we have been working on called EFFAA. Its scientific name is alpha-amyrin, which is available in fruits like passion fruit. So this molecule, we want to detect whether it's effective in reduce tau pathology using this 3D culture, ADiPSC-derived neurons, astrocytes, and with a rat microglia co-culture system. And this brain-in-a-chip model in the AD background showed very dramatic p-tau217, a marker for AD compared to the control group. And you can see the number 4 with our passion fruit molecular treatment, we can dramatically reduce p-tau217. As Shrasha mentioned, inflammation is another driver of Alzheimer's disease. And in this model, increased inflammation is evidenced by migration of red dotted microglia from the rim to the middle. The more in the middle indicating more inflammation. And our molecule was able to dramatically reduce inflammation. In addition, our molecule had a favored pharmacokinetics of 10.1 hour in the young mouse blood. Alpha-amyrin was also able to pass the blood-brain barrier. It was able to be detectable after 2 hours injection into the vein. Before the publication of this small molecule, we had a paper 4 years ago showing the crude extract of passion fruit was able to at least reduce memory loss in animal models. In humans, we didn't know. And we got news from the national TV channel of Norway. This was published on Friday. So when we went to the market on Saturday, and the passion fruit was gone, which indicating how important the science to drive the daily life of the general population. So I think to do aging research, like basic researcher like me, a clinician like Andrea Maier, as well as politicians in Singapore, in Norway, in many other countries, we need to work together to address the aging situation. And I was very fortunate to have like big wigs like Andrea Maier, among others, to write this review. We wanted to bring all us together at a molecular level to address aging at a molecular tissue level, as well as a clinician, what are the current challenges in the aging field, and how can we address those issues. And finally, at a societal level, how we're working with the politicians, with the lawyers to build an aging-friendly society. And I invite you guys to read this paper if you have time. We have the similar concept in Norway doing dementia research and aging research can't be done by one person. We need to be united. And we have built this Norwegian Center on Healthy Aging with our colleagues. We do aging at a cellular, individual, and societal level. We also have built a national level anti-Alzheimer's disease network with many of the colleagues who are our board members. Here, I want to give you a summary of my today's talk. So first, I explain to you why mitophagy is reduced during aging, especially in Alzheimer's disease. At least reduction of protein ULK1 is one of the causes. Genetic upregulation of mitophagy through ULK1 overexpression forestals Alzheimer's disease pathology in preclinical animal models, which enable us to conclude that defective mitophagy is a risk factor for AD, while mitophagy is a drug-able target. We also worked on different mitophagy inducers, such as uranexin A, which I didn't show you here, but we are now running a phase 2 clinical trial. Other mitophagy inducers like NAD+ precursor, as well as a passion fruit molecule alpha amyrin, also hold potential benefits, but with more clinical data needed. We also shared with you that NAD+ may not only benefit for dementia, but also benefit for a group of orphan disorders, premature aging, based on the success of a series of clinical trials we and our colleagues finished in Europe and in Japan. Here, I want to thank all our team members, collaborators, our major funding. And definitely, I would like to thank Andrea for this opportunity to speak again in your platform. It is my great honor. And I also welcome you to Oslo in August.
Thank you so much, Evandro. Beautiful talk. Let me start with a more general question for you. You are building these great networks within Scandinavia, in your own country, in Norway. What needs to happen to really drive change in your country you are living in?
Yeah, we had discussions with the leaders in our institutes and the university. We believe how to have a good communication with politicians could be the most efficient and effective way, including to have them to allocate more money for basic research, as well to invest more money to build more nursing homes, to change people's attitude of life, rather than to treat a disease, but to prevent a disease, which you are leading this in Singapore. I think what the work you are doing in Singapore also inspired myself and other colleagues to do in our own countries.
Thank you. However, how do you build the network between basic researchers, clinicians, and then bridging them to politicians? How do you build that?
I think I was very lucky after I finished my postdoc in 2017. Professor Wilhelm Bohr, introduced me to the Society of Norway. I think at the time I was very lucky to be able to, with networks, with the dean, with the president of the university, and with their wisdom and their networks, it helped me, a small potato, to reach those people and get the things done.
You're not a small potato. But good. Let me go a little bit into your field of research, and that's dementia and Alzheimer's disease, which are so prevalent. How much of AD pathology you think is mitophagy driven?
One answer could be, I think, 30 to 40%. So our answer was based on when we giving ULK1 treatment, when we giving mitophagy inducer treatment, we can dramatically reduce the percentage of A, beta, and tau, but cannot fully stop them. So compared with the disease group with vehicle treatment, and our mitophagy stimulation strategy can reduce 30 to 40% of the pathologies.
Well, that's a lot, because if you think about that, you could reduce the pathology by 30-40%, that's immense. Or is it the way that you have two different types of AD, that you have more the ones of mitophagy being driven and other mechanisms? So is it heterogeneity within individuals, within the mice, the humans, or are there potentially more mechanisms being involved?
I think you raised a key point, which is also the challenge in the Alzheimer's disease field. Also, as Shrasha mentioned, even we have named this disease by a German professor for more than 100 years. There are so many groups working on Alzheimer's disease, but still we do not have a humanized animal model for AD. The common used Alzheimer's disease models like APP, PS1, A-beta based, those are based on the familiar Alzheimer's disease genetic mutations, which only contribute to less than 5% of the AD. While the 95% or even more, we do not have a good model for them. And for ApoE4, which is a genetic risk factor, but for the mice, when they overexpress ApoE4, they do not show the good phenotype as that in Alzheimer's disease. So there's a species to human gap, which limits us on how much percentage of our data can be translated to humans. That is why in our group, in addition to using mice C. elegans to test our drug candidates, we are also more focused on using this brain organoid or brain on a chip system to mimic the human brain and see whether this compound can reduce any of the pathologies.
Let me be a little bit provocative. So what you are proposing is that Alzheimer's disease is a classical disease of aging, which is quite different from how it was perceived when I was in medical school or I was working as a geriatrician. Because there it was that specific disease, it's very AD specific and accumulation of plaques, etc. So how is it being perceived with neurologists, what you are doing?
We actually have very close collaborations of neurologists. So my lab is physically localized in one of the largest hospitals in Norway, Akershus University Hospital, and my office is 2 meters away from the neurology department. So we working together in running NAD-based clinical trials for ALS and PD. AD I'm not involved in. I think first there's a knowledge gap between neurologists to the basic researchers. Some believe, some who do not believe. But for many of the neurologists who are at the forefront of the research, I think they are convinced and they are also giving the voice that Alzheimer's disease is not just an isogenic disorder, but could be a group of disorders which have tau and A-beta pathologies. So it's still, I think, a very tough question, and I may not be able to address you very well.
But it is a disease of aging or a consequence of aging?
At least colleagues think aging is considered as a primary driver of Alzheimer's disease. Yeah.
Okay. We are negotiated, so yes, it's a consequence of aging. That's great.
I hope, yeah. I hope in the forthcoming fourth Norway UK meeting, because we are having a lot of clinicians and basic researchers, I'm very happy to see your opinion in that meeting in Oslo.
Good. But what you are driving is really the force towards its autophagy, especially mitophagy, might be a driver, be a solution for reducing the symptoms and reducing Alzheimer's disease. And of course, mitophagy and mitochondrial function is one of the hallmarks of aging. And what you're actually proposing is you're giving NAD precursors, you're giving urolithin A, you're giving extracts of passion fruit. You could actually turn it around, say, we are treating aging, and therewith we are also treating AD. So in your trials you are doing, are you also capturing more organ systems than just the brain in the trials to actually see how much systemic effect you could see with your trials?
Yeah, I think you raised a very important thing is whether those mitophagy inducer approach can also slow down the aging. Because of the ethical regulations and the demanding of the money for this phase 2 clinical trial of urolithin A to treat on AD, I think we only included the grape strength, this type of parameter, as a mark of frailty in. But we indeed are working intensively on the question you asked, whether those mitophagy approach can slow down the aging process. We have a paper in preparation. So at the time when we generated a ULK1 overexpressed mouse model to cross with Alzheimer's disease mouse model, we saw reduced pathology. When we are maintaining the colony of ULK1 mouse model with wild-type background, we saw those mice live longer, had less like gray hair compared to the wild-type mice of same age, which encouraged us to run a lifespan, health span in those ULK1 overexpressed mice. And we indeed saw over 20% increase of lifespan in both genders, while in females 30% and male 10% with 2 biological repeats. So definitely we believe mitophagy induction, at least in animal models, can extend the lifespan and reduce the aging process.
Yeah. So it might be very nice to also include a 6-minute walking test and quadriceps strength, where there are many publications now showcasing that either in animal models or in humans, urolithin A might have a good effect, and also the NAD precursors. And that leads me to another one, and that's alpha amyrin. So the passion fruit one. So if you want to buy passion fruit, I think around your university it will be difficult because everybody is grabbing it. Tell us, what is this molecule alpha amyrin, and how much of passion fruit might we have to consume to get to a level which might have a beneficial effect for our brain?
Yes, I think for this unlock aging conference you have been organizing, my senior postdoc Shu Qingcao was so pleased to be selected in your meeting to give a talk. And she had the idea to working on passion fruit 5 years ago, or even longer, when she was working in Chulalongkorn University in Thailand. Because in Thailand and many other countries, there are epidemiological studies showing consume colorful fruits and vegetables correlates with reduced chance to get dementia. And Shu wanted to know whether this is just a correlation, because those people having those fruits are also financially sufficient and get better access to medicine, or because there are still molecules inside. And we isolated more than 100 compounds in passion fruit and through one by one for most of them, and we saw alpha amyrin worked the best. It is a small compound with, I think, small phenol. I cannot recall it's belong to what type. Speaking how many passion fruit to eat to achieve the dose we can get for humans, we have been challenged by our colleagues to do a mathematics. But to the best of my knowledge, we were unable to do, because we haven't really figured out how much in the juice, how much in the seeds, and how much in the skin. But my normal suggestion is maybe 2 per day to start with, before we have more accurate answer to you.
Okay, thank you. Thank you so much. Highly appreciated. Let me ask you my last question before getting to the questions of the audience, and it's a massive amount. ULK1, would it be a good marker to introduce into screening, into memory clinics, to predict who might have MCI in a couple of years' time or dementia?
Yeah, so for this Nature Aging paper, I think it will be online in the coming 1 to 2 months. We indeed used both CSF and serum samples. So ULK1 was detectable in both serum and CSF, and it is correlated with AD continuum. So if our data can be repeated by any other big cohort, if they also saw the similar thing, then we'll be very happy to move this forward as a potential biomarker, or at least as supporting biomarker in addition to what we already have.
Okay, that's nice, because then you could even do head-to-head comparisons with other markers if it has additional value. Okay, over to Sharanya.
Thank you so much, Prof Maier, for your lovely introduction, and really enjoyed your presentation, Prof Fang-Stavem. Possibly because aging and Alzheimer's is also topics of interest for me as well. So there I felt a lot of common ground in a lot of your research questions as well. So one of the questions from the audience, which I think really pertains to, because we know NAD mechanisms, mitophagy, are all cell-related mechanisms as well. Are there also defects in other clearance mechanisms, such as proteasomes, that you have noticed in your work?
Definitely. We haven't done, but my colleagues have been shown that reduced UPS, ubiquitin proteasome system, which is another protein degradation system, is dampened in Alzheimer's disease. Very recently, we had a paper in Science Advances showing compromised mitochondrial UPR also plays a role in Alzheimer's disease, while NAD+ can increase mitochondrial UPR as well as increase RNA splicing as a way to reduce many of the side drivers of Alzheimer's disease.
Thank you so much. Another question from the audience, which I pertain to your work with multiple clinical models as well. All the NAD precursors, how much bioavailability in brains of humans and mice have you noticed? And is the benefits largely directed in the brain, or is it basically other indirect systemic effects? So can you shed any light on that, please?
Definitely. I think compared to other inducers, NAD has its own advantage, because NAD+ and its precursors are essential metabolites in our human body. So if we maintain the NAD level to our 20s, then theoretically it could be safer rather than you to consume other compounds isolated from plants or other resources. There has been a very I think intensive discussions, debates, or research on what is the best efficient route to give NAD+ precursors like NR and NAM, whether they can be degraded majorly through the microbiota if we consume directly, and whether and how many of them can pass the blood-brain barrier if to benefit for the brain. For the brain, my colleague in Bergen, they finished a phase 1b to phase 2a clinical trial in giving the NAD precursor NR for Parkinson's disease. Using the non-invasive, I think, MRI, they saw NR was able to dramatically increase the brain NAD. I think that is NAD, which can include NAD plus NADH, because that method may not be able to distinguish NAD plus and NADH. Whether the NAD-based benefit, for example, memory, is autonomous, means like brain dependent, or non-autonomous, meaning it can increase cardiovascular benefit to indirectly improve the brain. I think the answer could be very likely for both, because when we increase NAD to physiological level in those premature aging or old animals, we saw the benefit in other non-brain organs and systems also.
Thank you, Prof. Stavem, especially because, as we know, no system works in isolation. All biosystems works together. So, yes, possibly other mechanisms could help as well. Another question from the audience is related to the pathway. So, the ULK1 is primarily controlled by phosphorylation of AMPA-K and AMKK and mTOR. So, how does that change or impact ULK1 function in your the overexpressed model? And is it just the ULK1 sufficiency, or you need changes in phosphorylation as well?
Yes, that is a very interesting and important question. So our lab is now working on why ULK1 is reduced during aging, and especially in Alzheimer's disease. And also, once we generate a ULK1 overexpressed mouse model, how it changes in the whole system. Over the past 1 year, we collected hippocampus, muscle, as well as liver. We worked with our colleagues in Freiburg University in Switzerland to run general proteomics as well as phosphorylation. And the interesting thing was ULK1 overexpression does not change the general protein expression level too much, but it can dramatically change the phosphorylation of many of its downstream proteins as a way to regulate those protein-related pathways. And we are now digging into more details on what are the key downstream proteins which are phosphorylated by ULK1, and how those phosphorylation or activation of those downstream protein contribute to the brain benefit.
Another quite interesting question from the audience, I believe it, how much passion fruit should be consumed to get the benefit? But related to that, are there any other like colorful fruits? Because we have always heard this adage, colorful fruits means a healthy biosystem. So are there other fruits or molecules that you have also investigated and found they give benefits as well?
Yeah, I answer this question in two ways, because that was also the same thing Professor Andrea Maier asked. When we consume a pure molecule like uronidine A, for example, in our case is alpha amyrin. For uronidine A, we do clinical trial like 1 gram per day. So then we can do the similar calculation for the alpha amyrin, the passion fruit one. However, when we eat the passion fruit itself, it may have other small compounds has the benefit also. So when we do the calculation, the method could be a little bit different based on how accurate we can reach. This is number one. And number two is whether alpha amyrin is also available in other fruits and vegetables. And the answer is yes. In this recently published advanced science paper, we worked with our colleague in Cleveland clinic, Dr. Feishong Chen, who has built food omics platform. So we found that in red peppers, in broccoli, in some other common vegetables, they also contain relatively low levels of alpha amyrin. I think, to the best of our knowledge, alpha amyrin has a high level in passion fruit.
Thank you, Prof. Fang-Stavem, especially because the dosage when you relate to molecules from like vegetables compared to when you have a drug which is fully bioavailable, I guess there would be a lot of disparity between that. Just a question of interest, especially for me. I wondered, you have looked at the levels of inflammation and in your research studies. How much do you think it is because of, let's say, traditional inflammatory regulators like microglia or astrocytes, or is it something beyond that, could be interneurons or astro? So what is the proportion that you have found, which is the elements involved in neuroinflammation?
Yeah, I think Saranya, you are absolutely right. In the AD field, scientists are pay more and more attention on inflammation, not only in neuron, but astrocytes and microglia. Our studies show that, for example, when the mitochondria are broken, damaged, and cannot be efficiently eliminated by mitophagy, the releasing of mitochondrial DNA can initiate the C-gas steam pathway, which is a very hot topic and a driver of inflammation. In our 2019 Nature Neuroscience paper, we also showed that microglia-based NLRP3 caspase-1 interleukin-1 beta pathway was also dramatically increased in those AD models, while mitophagy induction was able to shut down this. There's another one very short. In Harvard School of Public Health, or in Harvard Medical School, colleagues show like the extra, like blood-based CD4 and especially CD8 positive T cells may enter into the brain through the leaking area or through the transporter and can cause huge inflammation. And this would be something you may also be interested to work on.
Yes, all these have also been terms and topics I've come across with my research in CCR5 as well. Maybe one last question could be, especially when we are talking about Alzheimer's, the earliest intervention is always the most effective. So like that, in your drug studies as well, have you found an age or like age range where interventions at that point would have the maximum benefit? And even beyond interventions, if you look at some biomarkers to assess Alzheimer's as well, have you found like which is the time frame which would be the most beneficial?
Understood. I think this a clinician would be giving you a better answer. But the key point I share with you, and also Professor Andrea Maier has been working very hardly in Singapore, that is to giving our knowledge to the general population, to tell them to do ahead before the disease reach us. And my background in bachelor and master are both preventive medicine in school public health. So definitely we believe prevent and slow down the development of disease is much more efficient rather than to treat a disease, especially its very late stage. And in our mouse model, we show normally 2 months old of the mice before they develop the pathology, like 5 months for the 5XAD. When we give mitophagy inducer, we can dramatically delay the memory loss from 5 months to 7 months. That was answer in mice.
Thank you. I believe yes, time is over. Yes.
Yes, and I think that concludes it very, very nicely. Start quite early, so eat healthily and do your exercise, maybe a little bit of fasting to induce mitophagy. And don't forget the passion fruits. Thank you so much, Professor Evandro Fang-Stavem. Thank you, Shranya, for the beautiful moderation of this webinar. Then, for you as listeners, and thank you so much for staying, there are a couple of announcements. We still have a few limited places left for the Healthy Longevity Talent Incubator, which is happening at the 29th of June to the 10th of July 2026. We are selecting 40 of the most engaging world leaders of the next generation to come to Singapore into the Talent Incubator, which is, I would say, a boot camp you will never forget. Secondly, in terms of announcement, there is a joint grant call with Luminex Multiplexing Innovation, and that's an award. Some of you might have gotten information that Luminex Multiplexing, the Innovation Award, is having a grant call with the HLTRP stem cell core facility in Thermo Fisher Scientific. So please have a look, and if you have samples you want to be analyzed, there might be an opportunity for you. The deadline is in 4 days, March 9th. Then the next Healthy Longevity webinar is on the 19th of March 2026, and the CEO and the co-founder of Sangro will be there, and that is Shantanu Kumar. So for now, I will leave you with a short teaser of the next episode. Take care.
Hi, I'm Dr. Weilan Wang. I'm working here as Senior Research Fellow at the NUS Academy for Healthy Longevity and also Healthy Longevity Translational Research Program at the NUS. Most of my research work focuses on the biomarkers of aging and also data analysis from data collected from the cohort studies and clinical trials. So instead of only focusing on our chronological age, my work is more focusing on how to assess the biological trajectory of aging in each individual participant.